Hello and welcome to this video on the genetic modification technique known as CRISPR. With the Philippines recently approving golden rice, the conspiracy theorists and mad hatters will come out of the woodwork to decry it as an unnecessary evil. These same people are why there have been more than 20 years of safety testing before this approval occurred. The delay in commercial release of golden rice is largely driven by ignorance and misunderstanding of what a GMO is and how the most common versions are achieved. Hopefully this video will help to address these 2 points in particular and provide a wider context as to its use in GMOs. GMOs, or genetically modified organisms, are a collection of life forms that have had genes changed via artificial means. Different states define artificial means as being certain techniques only. These could include viral vectors, plasmids, and hybridization of certain cells to create chimeras. It could explicitly exclude radiation-caused mutagenesis and conventional hybrids. The most common definition used in practice is modified in a way that does not occur naturally by mating and/or natural recombination. These distinctions are important when looking at GMOs. That is because CRISPR falls into a strange place in many respects, while it clearly fits into each category in other cases. As has been described in greater detail elsewhere, CRISPR, and the more ubiquitous Cas9 version of CRISPR, works through a bacterial immune system feature, which would normally cut foreign DNA into smaller pieces, which are then ejected from the cell. This would nominally keep the bacteria alive. By creating a targeting mechanism to direct the Cas9 mechanism, a gene can be cut out and replaced with something else. In other applications, it could add a gene into the sequence, but not necessarily remove the original. For more details, see the video linked in the top right and in the description below. This process is fundamental to why not all CRISPR-modified foods, and to a certain extent animals, are GMOs. In some cases, foods that have been genetically modified have not been modified in a way that fits this criteria. A good example of this is the modification of a gene that is a different version of an existing one. This could be to increase or decrease the expression. Think of what would happen if you were to increase the activity of a gene that responds to CO2 levels in a plant that lives in a CO2 sink, or as a response to climate change. It should grow better, but you have not made an unnatural change, as something similar could be achieved through hybridization or simply crossbreeding. Another is a hormone called gibberellin, which is needed for plant height to be achieved. This is modulated through a gene called GA20OX. This controls the amount of gibberellin produced in plants. Modifying this would change plant height, which in turn alters yield or use. Tall grasses for hay and short grass for lawn. This can be done by targeting that gene with the CRISPR toolkit and swapping, altering, or even adding genes. Another way that a CRISPR-modified organism might not be a GMO is one of inheritance. Once the originally modified crop passes on its gene to the next generation, it no longer fits the definition of a GMO. There was no unnatural inheritance of the genes, as it was all normal, a natural mechanism that has been observed for as long as reproduction of life has existed, and is especially evident in sexual reproduction. In 2018, the first of the GMO products were approved that followed under this particular line of reasoning. It was a particular canola variety, and more are being brought to market in 2019. The advantage to this process is that it avoids the problem of being a GMO, but also does not take decades to bring a new variety of a crop to market. Now let's apply this to the topic of golden rice. As per the Golden Rice Project website, the rice plant possessed the machinery needed to synthesize beta-carotene. Throughout the whole plant, this is a fully functional mechanism, but it is disabled within the grain itself, which is the part consumed, by adding two genes, one called the plant phytoene synthase and a second gene called phytoene desaturase. They were able to turn the beta-carotene genes back on inside the grain. This led to accumulation of beta-carotene and other vitamin A type products. However, these were only attainable at levels of 1.6 micrograms per gram, nowhere near enough to meet the daily vitamin A requirements. The second generation and modifications made looked at biosynthetic pathway, and in particular a rate-limiting step that controlled the movement of the whole pathway. Examination of the synthesis pathway identified the plant phytoene synthase gene as being responsible for rate-limiting the synthesis of beta-carotene and other vitamin A possible products. They were able to then look at what was happening here, compare it to other species, and see what could be taken from these other plants and put into rice to make it produce more vitamin A or provitamin A products. This led to the identification of a version of the plant phytoene synthase gene in maize that could be transferred to the rice. Doing so produced the second generation of golden rice, sometimes referred to as GR2, which produces 31 grams of beta-carotene and 37 micrograms per gram of the carotenoid group products. This is distinctly a GMO in Generation 1, but in Generation 2, which is produced from the currently approved crop, it may go from a label that says GMO to just being another agricultural plant, one that has inherited its genes for a vitamin. This is an important milestone, with more crops coming to market as a genetically modified organism. It will be a question of which are now considered a GMO and which are not. This is important for a simple but fundamental reason: mortality and morbidity. In the Philippines, a universal source of vitamin A will reduce childhood mortality by 23 to 34%. It will be responsible for decreasing measles cases by 50% due to the immune modulating effect of vitamin A. In the Philippines, this is a massive change and could lead to a huge improvement in the quality of life and the sheer number of people alive due to vitamin A deficiencies. GMO, as it is used now, is almost a nebulous term in regards to what is being discussed in approvals processes. The specific details it aligns with are mitigated, if not outright transient, when we apply a consistent standard. This is a result of trying to define a GMO, but nothing we do, even CRISPR, is not going to fit this criteria after 1 or 2 generations. If we were to try and apply a rule to GMO crops that use CRISPR, then we would quickly have to expand it to include more conventional modification techniques like hybridization, crossbreeding, or even radiation mutagenesis. Use of the GMO label is ultimately nothing more than a mark of shame that is being ignored more and more frequently as people are educated on the meaning of it and what actually happens with a GMO. This leads to a realization that it is nothing more than what we as a species have done for millennia, but with a more refined mechanism and predictable results. Thank you for watching this video. If you have found it interesting, please consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.